People are joining, so I will give everyone like a minute or two, and then we can get started. Okay, so I think that we can get started. Thank you, everyone, for joining, and thank you, Gabriel, for willing to give this talk. So we have today Gabriel Piedrafita. Gabriel is a biochemist. He studied in the Complutense University, and then his thesis in theoretical models for the biology, I guess. And after that, he did a postdoc at the Sanger Institute in the UK, and then came back to work at Theneo for a few years. They're working on clonal competitions. And now he is an associate professor at the Complutense University. And he's going to be talking about inference of somatic mutations from single cell transcriptomics survey in the esophagus. So thank you so much. The floor is yours, Gabriel.
Thank you. Thank you very much, Anna. Good afternoon, everyone. So as Anna said, in our group, we are interested in somatic clonal dynamics in physiologically normal tissues. And I will tell you why in a second. What I'm going to present today has to do with a side project in our group, and has to do with how or whether we can infer somatic mutations from single-cell transcriptomics. So why are we interested in physiologically normal tissues? This is because mutagenesis is not exclusive of cancer. No? So we have typically devised cancer as an evolutionary disease that progresses with the acquisition of mutations. Once a particular mutation hits a cancer driver, it causes a series of clonal expansions that ultimately lead to cancer. However, in the last few years, we've experienced a plethora of publications describing how mutations are not exclusive of cancer, but they do accumulate as we age. And this occurs in clonal hematopoiesis, but also in fast-renewing tissues such as the skin or the esophagus, and even tissues that are long believed to be quiescent, like the brain or the bladder. So I'd like to raise this analogy. So here we have like a fern field, and fern is a very rapid colonizer. However, it does not typically form huge trees. So we can ask what the phenotypes of those mutant clones arising in physiologically normal tissues are, and also what their role is in carcinogenesis. So to address this question, let me show you two examples of how cell competition matters. So here we have an example from this publication where authors studied by deep sequencing the mutational burden in single cells in human lung epithelium. And intriguingly, what they observe is that they have current smokers where cells keep very large, very high levels of mutations. You have here the histograms in purple. However, what is interesting is that in ex-smokers, there's a bimodality distribution in the frequency of mutations per cell, indicating that there are certain subpopulations of cells that keep high levels of mutations, similar to current smokers. However, there are small populations of cells that keep small levels of mutations, similar to the ones from never smokers. So the authors suggest that most likely these subpopulations of cells that are more protected from mutations might be due to quiescent stem cells that ultimately replenish the tissue and are able to outcompete mutagenized cells. In the esophagus, and this is a dramatic case, as we age, we accumulate myriads of mutations in cancer-related drivers, such as TP53 or NOTCH1. And what this means is that by middle age, more than half of our esophagus becomes colonized by one of these mutations. So the question is, what is the phenotypic effect of those mutations? Or in other words, if we were to live longer, would we expect to develop cancer, or these mutants somehow, their phenotype tempers down? So this is a study, I didn't say this is a study by Inigo Martin Corena and colleagues, where they perform ultra-deep targeted DNA sequencing on 1 centimeter square biopsies from healthy donors. And as you see, by 72 to 75 years old, most of the epithelium is colonized by cancer-related mutants, raising again the important question of where those mutations are going, whether they would relentlessly form tumors given enough time or not. To address this type of questions, we typically use mouse models in our group, and we do what we call lineage tracing. How does lineage tracing work? What we do is we work with transgenic mice that upon tamoxifen administration, they express a fluorescent reporter. And this fluorescent reporter can be linked to a particular point mutations, such as this one in P53. So by doing so, once we give a certain dose of tamoxifen, we are able to follow individual P53 mutant clones over time. What we do is we recast statistical properties from clone sizes from all these groups of P53 mutant clones over time, and we fit these distributions of clone sizes with different theoretical models. By doing so with different mutants, not only P53, but also mutants in Notch pathways and others, we've observed that most of these driver mutations, what they exploit is a phenotype where they gain advantage by differentiating less than they should. So it's not that these mutants, they divide faster than wild-type cells, but what they do is they differentiate less than wild-type cells. So they remain basal. And in these tissues, basal means proliferative. Differentiated means they have exited the cell cycle. Why is this important? In another experiment, ex-colleagues from the Sanger Institute, what they did was they treated mouse models with DEN. DEN is a nitrosamine, and it causes mutations. So after this DEN treatment, they induced animals to track individual clones over time, following the lineage tracing principles that I've been speaking about. So they had two cohorts of mice that were controlled and mice that were treated with them. The first obvious thing that we observe is that clones get much larger but are much fewer over time under control conditions. But what is important is that those mice that were treated with the mutagen developed much larger clones and were much fewer. And this is an indication of selection. Indeed, we perform deep sequencing, deep DNA sequencing at the ultimate time point, and we observe how most of the epithelium, if not virtually all the epithelium, gets colonized by mutations in Notch pathway. However, the tissue remains almost devoid of any tumors. How is this possible? So we have selection for certain mutants, however, they do not develop tumors. Again, we calculated clone size distributions from all these clonal data, and you have here them displayed in a funny fashion. I'm not gonna go into details of how we do this. The thing is that we perform simulations to fit the experimental data, and what we observe is that quantitative data is consistent with a scenario where mutants, they get selected over time at intermediate periods of time. However, as time goes by, this level of selection resolves, and these mutants somehow they temper down their phenotypes, and the tissue remains homeostatic, meaning that we have mutations that acquire certain advantages, but however, they go back to normal behavior. They do not develop tumors. And why is this so important? We can think about a scenario of carcinogenesis. And in fact, these mice, at some point, in some particular places, and this is a peeled esophageal epithelium, you can observe how under certain regions we observe some localized tumors that stain positive for keratin 6. So some of these mutants, they are rapid colonizers like the fern, and they develop in the normal areas of epithelium, and they have the ability to outcompete and eliminate the tumors in mice, meaning that resolving the mutant phenotypes, not only in humans, but also in mice, is key to understand how carcinogenesis can be preempted. Okay, so how can we infer mutant phenotypes in humans? Because obviously this is all lineage tracing in mice. So people, and this is a very recent report that was published in December, what they did was to co-capture DNA with RNA at single-cell resolution to be able to map genotype to phenotypes in humans. However, this is far too expensive, and this is something that cannot be afforded by most our groups. So what can we do? We cannot do lineage tracing. This is far too expensive. Very recently, and in the last couple of years, there's been different methods suggesting that it is possible to detect mutations exclusively from single-cell RNA sequencing. And this is a very good and very tempting method because we have many data sets published, for instance, from the Human Cell Atlas, and we could leverage all that information potentially to detect mutations from all those public data sets. So this is a publication from Francesc Mullas et al., published in Nature Biotechnology, and this is one of the most important methods that has been published to detect mutations or that tries to detect mutations de novo from single-cell RNA sequencing. What I mean with de novo means that they do so without relying on any DNA sequencing tool to build a germline reference. How does schematic pipeline works? So what they do in the publication is, first of all, they start from a typical single-cell RNA sequencing experiment. By the way, they can do so as well with single-cell attack sequencing. And they use the aligned BAM file, and they split the BAM file into cell type specific BAM files once we have already annotated individual cell populations. Secondly, it performs variant calling, and it does so by cell type comparison. So basically, for any position in the genome, it compares the allele of cells for any cell type versus any other. And third, it performs a beta binomial test to basically retrieve significant mutations. And after applying a set of hard filters, they end up with a final call set of somatic mutations that they present in a TSV file. So we took advantage of a schematic. And before explaining how we proceeded, it's important to mention how authors of the schematic developed this tool, but it's important to notice that schematic requires alternative and reference allele represented in different single-cell transcriptomic clusters, meaning that the tool is most suited when we want to compare a cancer cluster versus a normal cluster. However, how can we work with a schematic when we have a polyclonal tissue, such as the ones that we've been describing? How can we tweak the parameters? This is something that has to be worked on. So the objective of this project was to address whether we could exploit single-cell RNA sequencing data to study the phenotype of mutant clones arising in normal esophagus during aging. To do so, the first thing we did was to validate whether single-cell RNA sequencing technologies are suitable to to reliably detect mutations. And this is work that has been done in collaboration with Maria Alcolea and Greta at the University of Cambridge. And what I'm going to present today has been the results of analysis from Alp, a PhD student in the group, and Dario, a former master student. So I'm extremely thankful to them. So first thing we did was we took single-cell RNA sequencing data from mice models from Maria and Greta. And Maria and Greta had animal colonies that had been treated with the nitrosamine that I've been talking about, and compared to old mice or even young mice. They had 8 libraries that were submitted to 10x Genomics, single-cell RNA sequencing. The interesting point here is that in this experimental dataset, they performed sequencing twice on every library. So every library was sequenced in 2 sequencing sessions. First thing we did with their data was to annotate the populations. Well, we filtered out non-epithelial populations. We filtered out epithelial cells and epithelial populations that have a high content of mitochondrial genes and were thus bad quality. And finally, we end up with this data set where we find 5 clusters of epithelial cells, 4 of which were highly conserved no matter what the condition of the animals was. So they were present in both young, old mice, and den mice. However, we have a 5th cluster that was exclusive of den treated animals. And we explored further this cluster, and we observed that it was enriched in keratin 6A and in keratin 14. Keratin 6A is a protein and is an RNA that is highly associated with incipient tumors in this animal model. So we annotated this cluster as a tumor-exclusive cluster. And this is important because in this way, we can make sure that our dataset is suitable to explore cellular phenotypes. We have a differentiation axis that goes from these 3 clusters down towards the suprabasal cluster. And we have a cluster of cells that can be mapped to tumors. Okay, now we try to apply a schematic. We transformed our BAM files and joined all the information from the BAM files and ran schematic, obtaining results from a schematic in a TSV format. By doing so with the typical parameters, default parameters from schematic, we ended up with 8,000 putative variants. However, how reliable are those variants? So first thing we did was to analyze whether those variants were specific of a single library or whether they were spread across different animals. And interestingly, most of the variants that we originally detected with the schematic were indeed spread through multiple libraries, indicating that they most likely correspond to errors or maybe to germline polymorphisms that are shared among littermates. In fact, when we represent the variant allele cell fraction, that means how many cells contain those variants, we observe that variants that are spread across multiple libraries are typically occurring in many, many cells, indicating that most likely they correspond to SNPs and not to somatic mutations. So we excluded the multiple library mutations. And then, secondly, what we observed is that from those mutations that were exclusive of one library, many of them fell into repetitive or low complexity regions of the genome according to repeat masker, and they were filtered down. We kept the non-repetitive variants, and from those, we had a very few mutations that occurred also in many cells in the library. So those were filtered out as they might be putatively germline variants. And we ended up with a list of 900 somatic mutations or variants that we can trust as somatic. Bear in mind that this is almost 10% of all the variants that were given by schematic initially, meaning that filtering is key for these methodologies. When we analyze these 900 putative mutations, indeed we corroborated how the mutational burden was more than twice higher in then condition compared to young or old, indicating that to some extent we can reliably detect some mutations with this methodology. However, half of the mutations that we detect might still be spurious. Understanding how somatic mutations behave under these conditions does not conclude here. So we have detected the variants, but how to characterize them? This is something that hasn't been included in schematic. So we have to customize Python scripts to be able to map those mutations to the barcodes, and finally to annotate those mutants with BEP to basically to be able to study their functional role. So, and these are the results of the mutant characterizations that we did using these customizations. The first interesting thing is how do the mutations compare when we analyze the two different sequencing runs, the two different sequencing batches. And this was striking how most of the mutations were exclusively found in one sequencing session or the other. Only very few, only 14 point mutations were observed in both sequencing runs. We speculated that this could be due to the very limited read depth that is achieved by single-cell RNA sequencing. And indeed, when we analyzed the read depth for the variants observed in unique sessions versus shared sessions, we observed that the read depth was extremely low and just above the limit of detection, indicating that indeed the reason why we have very few mutations that are shared between sequencing sessions is a matter of read depth, of very limited read depth. And this would ultimately limit our sensitivity when detecting mutations from single-cell RNA sequencing. A second aspect that is also important is the cellular representativeness. When we analyzed the barcodes from both sequencing sessions, we were able to retrieve almost all cellular barcodes in one versus the other sequencing session, indicating that indicating that it's not a matter of cellular sampling after the library construction. However, there are strong cellular sampling limitations before library construction. When we plotted the apparent clone sizes from those mutations, we observe that most clones that we observe with this technique have very few cells, from 2 to typically to 10 cells maximum, indicating that most likely most of the clonal cells are not captured during the cellular isolation, and only very few enter into the library and are finally sequenced. This level of sparseness somehow explains why, when we address and when we question what the genes in this panel are mutated, we observe that typically most of the mutations occur in genes that are non-cancer related. For instance, most of the most prevalent genes were involved in housekeeping functions, such as here we have ubiquitin or USP10 that are involved in ubiquitination, but we have also examples of other proteins that are high expressed and they are involved in mRNA splicing or energy metabolism, indicating again that what we are detecting is most likely passengers and not that much drivers. Indeed, when we explored the impact from those mutations, the impact given from VEP, most of the mutations were mapped as modifier or were characterized as low impact. And we had very few high impact mutations. Still, we did the effort to map where those mutant barcodes mapped into the UMAP embedding, and we found some intriguing and some interesting cases. We have here cases of high impact variants affecting cancer-related genes under the DEN condition, and these clones are mostly basal. And also we have some cases of missense variants in other cancer-related genes in the control mice. Here, ITGA3 or 3 other clones. Not only that, we also found some clones that were exclusively mapping to the DEN cluster, exclusively mapping to the tumor-related cluster. And these are 2 mutations in DST. DST encodes for dystonin, and dystonin loss of function mutations in dystonin cause epidermolysis bullosa. And also, we found a mutation in STFA3, and this encodes cystatin A, and cystatin A is a key regulator of keratinocyte differentiation, which is upregulating in some skin neoplasms. So all this makes sense. However, as you can see, we have limited evidence of driver clonal expansions from this data. And now we're going to switch to humans. Now that we've observed some of the limitations of these methods in mice, basically limited read depth, plus the question or the problem of cellular sampling, let's see whether we can characterize mutations from human single-cell RNA sequencing data. The problem with human single-cell RNA sequencing data is that using schematic tools, it is very difficult to build a reference genome, because previously in the data that I've shown, we could build the reference allele based on the littermates. However, how can we proceed with humans where every individual has its own SNPs? So we took advantage of this paper, and this was published in 2020, is part of the Human Cell Atlas, and they did a single-cell RNA sequencing experiment that had the interesting property that they did sequence single cells separately in 4 adjacent regions of tissue. So we took advantage of this multi-site design to build reference alleles. How? So we would consider each site, and we would consider other regions from same donor to be reliably contributing to make the reference. So taking data from this publication in esophagus, we annotated the populations and restricted to the epithelial cells. When we restrict to epithelial cells, we can observe how the data adequately represents the differentiation trajectories from most basal cells that are here at the bottom towards more differentiated, terminally differentiated cells here at this tip. And we can do so by RNA velocity here or by a pseudotime, like slingshot here. In this way, we can conclude how this dataset is also suitable to make phenotypic explorations. Now, what about the genotype? So we used schematic again, and as I said, we divided the dataset in cell type segment exclusive BAM files. We considered all these properties and we ran schematic, and we ended up with more than 80,000 putative variants. When we apply a repeat masker, and this is something that is somehow recommended in schematic, but it's not mandatory, and here we can see how it becomes key. The number reduces to more than 20,000 mutations. When we analyze those mutations, we observe how many of them are still shared between different donors, indicating that there's still much room for improvement, because most likely somatic mutations should be restrained to a single individual, unless they are very recurrent point mutations. So from these 20,000 mutations, we plotted how many of them were present in one segment versus multiple segments, be them exclusively in epithelial cell types or stromal cell types. What we observe is that most of these putative variants, they were supported by multiple cell type segments, suggesting that they most likely correspond to germline polymorphisms. Indeed, when we plotted the CCF that represents the fraction of cells that are mutated, we observe that those mutants that were detected in multiple cell types or multiple segments were observed in a huge fraction of cells, further suggesting that they are SNPs. However, mutants that are restrained to a single territory have more constrained CCF values. In addition, when we observe those mutants that appear in two segments, we observe many co-occurrences between different cell lineages, such as epithelial lineage, or a gland lineage, or endothelial, or other stromal populations. And these are unlikely to be somatic mutations unless they occur very early in development. So we reasoned that we would restrain mutations to keep only those that occur in a single segment or maximum two segments, but with a very limited CCF. When we applied those filters, we obtained a matrix of co-occurrences when most of our putative mutations are indeed epithelial in origin, meaning that our filtering steps have been okay. However, as you can see, we end up with 1,000 putative mutations from the more than 80,000 variants that schematic would give without any filtering. When we do so, we can have another validation tool that is to assess how many of these mutations are shared between individual donors. I explained at the beginning how many of the initial numbers are shared. However, with the final figure, only 30 putative mutations are shared between individual donors, which is more congruent with what we would expect. Once we have filtered our putative mutations, it was time to characterize them. In the case of the humans, we couldn't observe a mutational burden increasing with age, with the age of the donor, and we speculate that this can be due with the diverse exposures that these individuals have. The second aspect that was interesting is that we observe few mutations in cancer drivers. Still, we could observe missense mutations in these cancer-related genes. Bear in mind that we have, for instance, NOTCH2 that is highly prevalent in humans, NOTCH3, KRAS, but we didn't observe mutations in TP53 or NOTCH1 that are among the most mutated in human esophageal epithelium. So we interrogated why is this happening, and here you can see the overlap between the genes that appear mutated in our analysis versus genes that are cataloged as drivers in cancer and genes that were detected by Martin Corena and colleagues in their paper that I mentioned before. And you can see how the overlap is very thin. So we speculated that perhaps the reason for this is the limited expression of common cancer drivers. Indeed, when we plotted the gene expression values for most of these common cancer genes in the Martin Corena panel, we observed how they have gene levels that were much below the mean expression values in our dataset. When we compare the expression of those cancer-related genes to the genes that were mutated in our analysis, we observe how they saw much lower, much more limited gene expression values. And this might explain why we don't observe that many mutations in those genes. Secondly, we observe, again, as in mice, how most predominant mutations are modifier type or low impact. And this is in agreement with the very small apparent clone sizes that we observe and a reflect of the sparseness in clonal sampling when making the library pools. And finally, something that we could do, as we did in mice, was to map those mutated barcodes back into the UMaps. And still, we observed some interesting cases. Here we have a CDH1, which encodes for E-cadherin. And E-cadherin is a key regulator of keratin differentiation. And here we observe a clone that is mutated with a missense mutation in CDH1 and is predominantly basal. And also, we observed a clone that was mutated in MAP kinase. MAP kinase, again, plays a key role in keratinocyte and in general in epithelial differentiation. And here we have a relatively large clone. So we might speculate that perhaps those mutations are having certain impact and might be some drivers. Still, as I say, the evidence is very, very low. We have very few cases of putative driver mutants from this analysis. And the last thing that we tried to do was to study the mutant structure. So if we have here the grid, as we said, all these individuals were sequenced in a grid with 4 biopsies. So we might expect mutations to occur in a single grid, in a single biopsy, or maximum 2 biopsies because we imposed that cutoff. However, what is the rate of co-occurrence between those mutations? What we observe is that most of those mutants, they occur in isolation. However, we observe some cases of clones that were sharing cells between different mutations. And here we have one case of cells that acquire XRN2 mutations. And part of this clone contains as well mutations in ZNF691. However, this type of subclonal inferences can be extended to some other cases, and here we have another one. However, the sparse clonal sampling that I mentioned above limits our capacity to make inferences about subclones. Still, what we observe is that most mutations that contain co-occurring cells, they are restrained to neighboring regions in this diagram, to neighboring biopsies. And there are very few cases of these clusters that occupy more than 2 regions, indicating that somehow we are preserving the subclonal structure in the tissue. And this is a further evidence that these clones, even if they most likely are passengers, we can somehow trust that many of them are real mutations. So to conclude, we have implemented de novo mutant detection for single-cell RNA sequencing in the esophagus, and we observe that these methodologies require a significant level of customization. We need to apply adequate filtering steps to remove the large excess of repetitive site calls and also germline variants so as to study somatic mutations. And this is even more dramatic in normal polyclonal tissues. So the parameters, we have to play with the schematic parameters very deeply. The second aspect is that the low average read depth that is inherent in single-cell RNA sequencing data limits sensitivity to detect somatic mutations. And this is particularly dramatic in the case of cancer-related genes that typically express at low levels. Third, single-cell RNA sequencing data is suitable for rough mutant phenotype explorations. However, a sparse cellular sampling limits full clonal identification. And most likely, most of the mutations that we observe, therefore, are passengers rather than drivers. And finally, I'd like to include this conclusion, that is that future studies aimed at exploring somatic mutations from single-cell RNA sequencing should rely on complex experimental designs, such as the ones that we had in mind, that were either complementary sequencing sessions or multiple biopsies per donor. And obviously, we should address much higher sequencing depth to reliably detect mutations from single-cell RNA sequencing data, despite previous publications showing how we could exploit single-cell RNA sequencing for mutant discovery. So there's much room still for improvement, especially from the experimental side, but also from the computational side. We have to be very careful with the filters and the parameters. And finally, I'd like to thank people in the group, especially Alba and Dario, for their computational studies, and also our collaborators, Maria and Greta, for the single-cell RNA sequencing data in mice, and other collaborators, and also our funding. I'm happy to take questions.
Okay, thank you so much for the presentation and for the insights on how to deal with this complicated data. So we have already some questions on the chat, so I will read them aloud, and if anybody else wants to add the questions there, please do that. And I start with the questions by Nitin. Have you observed an effect of cell number on somatic mutation prediction? For example, in the human esophagus dataset, a higher number of mutations are detected in the epithelial compartment. Is this observation normalized for differences in cell numbers across cell types?
If you can repeat, please. I'm not sure if I understood correctly.
Yeah, so basically, different cell types will have different numbers of cells in the data sets. So the question is, if the cell numbers or the number of cells of a particular cell type impacts your ability to detect the somatic mutations.
Yeah, so if I understood correctly, so the question is whether the epithelial compartment shows higher clonal sizes as compared to stromal populations. This is in part the question?
Yeah, if there is a relationship between the number of cells of a particular cell type with your ability to detect the somatic mutation.
Oh, now I understand. This is an interesting question. This is something that we haven't addressed. Because we restrain to epithelial cells. In our data set, most of the, I'm gonna try to go down. In the original data set, most of the cells are epithelial in origin, and schematic, it performs variant calling at cell type level. So in principle, you should be able to perform variant calling for any given lineage. However, if you dig deeper, the variant color depends on how many instances of the reference you have. So it will more reliably work on populations that are larger rather than small ones. So I wouldn't trust variants that come out from other lineages that, especially, I don't know, B cells or so, that have few cells.
Yeah, okay, thanks. So another question by Nitin is that gene expression appears to influence somatic mutation detection. Do you expect this effect to be absent or reduced in single-cell ATAC data, given that it measures chromatin accessibility rather than transcriptional output?
We haven't explored single-cell RNA, single-cell ATAC sequencing data. I've heard some people that have been testing variant calling from single-cell ATAC sequencing data, and they believe that it could work better, but we haven't tested.
Haven't tried that yet. Okay.
No, we haven't tried.
Yeah, okay.
And it's worth mentioning that in our case we tested only single-cell RNA sequencing data from 10x genomics. So this is 3' based, not full transcript. However, in the original publication from schematic authors, they did also work on 10x. So
I see. Okay.
it goes in line.
All right. I switch now to Lara. So she thanks you for the very nice and inspiring presentation. Would the copy number assessment help to elucidate clonality?
Yeah, this is a very good point. Yeah, and we haven't tested. We probably should. Copy number variation is gonna give you a broader perspective on clonality. Meaning that, and probably in the case of polyclonal mutations, or in the case of physiologically normal tissues, it's not gonna help, because physiologically normal tissues acquire point mutations, but typically haven't got many copy number variations. However, this might be useful in the context of cancer. However, the level of resolution that you can achieve with copy number, maybe you are able to distinguish between 2 or 3 subclones in your tumor. I don't believe you can go much deeper. So
Okay.
yeah, the answer is yes. However, with limited subclonal resolution.
Yeah. Okay. Understood. Another question by Nitin was, how do you generate reference alleles for mutation calling when region-specific reference data are not available? Are there established strategies to address this limitation?
When we haven't got like a reference genome for
well, region-specific reference data, I guess that this is a reference genome of, I don't know if for some regions of the genome you don't have this information. But yeah, when this information is not available.
I'm not sure because I wasn't the developer, so yeah, I don't feel confident responding.
Yeah, that will be because it
question.
will be difficult to differentiate from
It will be
alleles, right? Yeah.
it will be very difficult. Yeah.
Okay. I have myself a question because I was looking at how much your filtering strategies impact or reduce the number of calls. Let's call it this way. So that means that basically out of the sequencing data alone, it's super noisy, the signal that you can extract, and then you need to start imposing these filters to reduce and to believe and so forth. But I mean, I don't know if you have done the math, but I guess that at some point you have enough of this noisy signal. Even if you impose filters, you will restrict the numbers, but maybe these calls that you obtain after the filtering are still, a random noise of somehow. So how do you have a strategy for validation of those after the filtering, of how you make sure that the filtering is not just a reducing in the number, but actually, giving you the right signal?
Yeah, that's a key question. And that's something that we've been thinking about a lot, both with the mouse data as well as the human. In the case of the human, as I explained, the first thing that we can do is we can take these parameters, such as the fraction of cells that appear mutated, and this is a first element of validation. So we observe that variants that we are keeping that are restrained to a single territory typically appear in a less number of cells. Also, when we keep them, we observe that clonal sizes, and this is something that I don't show here, but we observe how they tend to have also smaller clonal sizes. Also, we observe how by doing this filtering, this is a second element of confidence, or second element of pseudo validation, if we want. We observe how we enrich in purely epithelial.
I see.
So in clones that are restrained to epithelial barcodes. So again, we have an improvement, at least an improvement. I'm confident. I'm pretty sure we have still many variants that might be technical, might be spurious, and there's no easy way to remove those unless you properly validate by, I don't know, PCR and then or sequencing or yeah. And then the second aspect that we can, or the third aspect that we can bear in mind is how many of those putative variants map to other individuals. Because if they are common errors, or if they are mapping to regions where the reference is not well built, you could always argue that no matter how much you filter, you will have a high proportion of them that map to multiple donors. However, we didn't restrict by the donor occupancy, we didn't restrict by this fact. So by applying our filters, we observe how, in the end, our list of 1,000 mutations contains mutations that are almost exclusive of a single donor are not spread in through multiple donors. So without using this as a filtering step, we can in fact validate how very few of them, these 30 variants, are shared. So most of them are unique. I believe that most of them might be passengers, still mutations, but passengers. So having very little phenotypic impact.
Okay. So any other questions from YouTube? Sarah, you were also looking into that?
Yeah, no questions for now.
No questions? Okay. So if there are no further questions, then I would say thank you very much for this interesting presentation, and good luck with future work, and thank you everyone from participating today.
Thank you.
Thank you. Bye-bye.
Genial.